First, I've been asked to tell all the speakers from yesterday and today to stay in the room at the end of this session for a picture, a photo all together. And then I shall also remind all the people attending online to ask questions via the Q&A section on Zoom. And now our first speaker is Ali Punjani from Structura. Thank you.
You can hear me okay? Yeah, great. All right. Hello, my name is Ali. I'm the CEO at Structura Bio. We're the company that builds CryoSPARC. Very excited to be speaking to you today about new developments for single particle analysis and data processing specifically. And before I start, I'd just like to thank the organizers very much for the invitation to speak. So for some history, we released the first versions of CryoSPARC nearly a decade ago after developing some useful new algorithms for ab initio reconstruction and accelerated refinement in the computer science department at the University of Toronto. And earlier this year, we released the latest version, V5. Over that time, our team has grown and we've continuously worked on new methods and algorithms, some of which have become standards within the community, high performance implementations, typically on GPU, building up a robust software system for the back end of Cryo-EM processing, streamlined and powerful and hopefully clear user interfaces, and also real-time processing through CryoSPARC Live at microscopes. CryoSPARC and CryoSPARC Live are free for academic use. And the number of groups that are doing Cryo-EM, the number of Cryo-EM structures that are being solved continue to grow rapidly. And we're really thrilled and very fortunate that CryoSPARC today is one of the most widely used software systems for single particle processing. And that's very exciting. But the reason we've been working hard on CryoSPARC and continue to develop it further is really because, just like everyone here, we hope to contribute to the journey of structural biology to help visualize the machinery of life so that new discoveries and new drugs can be made that help people. With that in mind, we're really laser focused on the goal of doing our part to help make Cryo-EM really work well in practice to be fast, powerful, easy to learn, and so that more people can use it and more biological questions can be answered. Everything I'm presenting today is on behalf of the CryoSPARC team that develops, builds, and deploys CryoSPARC. So single particle analysis has come a long way, but of course there are still difficult aspects that show up in the data processing stage and start earlier. Sample prep, of course, remains a substantial challenge. Denaturation, air-water interface effects, orientation distributions, the amount of sample that's needed. Data collection has become much more streamlined and now yields so much data every day. Sometimes it's too much data to manage, and not all that data is always useful at the end. For answering larger biological questions, often multiple data sets are needed. Data processing has required sometimes expert knowledge, manual iteration, and feedback to reach state-of-the-art results. For the past couple of years, we've been working on some new developments to try and make single particle processing more as robust as possible to some of these challenges, and to be able to ingest real-life data that may be of variable quality, hasn't been manually curated, and still do a good job with it to get 3D density maps, and even to automate processing in certain cases. So I'm going to talk about those things today, starting with some improvements in pre-processing. The first one is CryoSPARC's micrograph denoiser method. This was released a little while ago, but we found it to be actually even more effective than we initially thought, so I just wanted to emphasize it. There have been many projects in the field working on micrograph denoising, generally based on the noise-to-noise concept, so things like warp, topaz denoise, etc. CryoSPARC denoiser is also based on noise-to-noise, with a couple of additions. The CTF is accounted for during training, making micrographs appear similar even if they have very different defocus values. We also found that combining a classical computer vision denoising technique known as total variation regularization helps to speed up convergence and gives higher quality results with smoother backgrounds. The denoiser is super fast to train, totally unsupervised, takes about 3 to 5 minutes on a new data set, only needs about 100 micrographs to train. And what it does is it learns to emphasize repeated particle structure. So after denoising, particle picking, even with the simplest particle pickers, becomes very easy. It becomes easier to separate particles from background. And picking scores, most importantly, become consistent across the data set, across defocus values, and even across different data sets. So highly recommended to try out the denoiser if you haven't already. Just for a few examples, this is a raw micrograph, a filtered micrograph, so just low pass filtering, and the denoised micrograph. Another example, raw, filtered, and denoised. On top of that, the next new development that's particularly helpful for challenging data sets, which was released more recently in CryoSPARC 4.7, is the micrograph junk detector. It's a neural network pre-trained that provides a semantic segmentation of a micrograph. We put together, to actually make this work, we put together an internal manually labeled data set, took quite some time to label, and split up different kinds of micrograph level junk into 4 common categories. Carbon and gold, so that's whole edges typically, intrinsic ice issues, so cracks, devitrified ice, graphene wrinkles, things like that. Extrinsic ice issues, so ethane, transfer ice, hexagonal ice, and then non-physical issues, things like beam fringes, gold reflections, residual drift. So the junk detector can detect these categories. It's trained from scratch, and that trained model is included in CryoSPARC, ready to go on new data sets. It's very fast. It's over 150 micrographs per minute. And you can use it to reject particles that are too close to junk. It also provides data set level statistics across a new collection of how much of different kinds of junk is present. This works quite well across data sets, really without any further training. Just a few more examples. And more recently, one project we started working on, this is not yet released in CryoSPARC, but coming soon, is to look into the effect of micrograph junk on pre-processing steps and other downstream tasks. So for example, carbon, gold, and ice, and their effect on CTF estimation. The presence of those junk things in micrographs can actually mess up all those downstream tasks. And on a typical clean micrograph, CryoSPARC's patch CTF estimation produces a defocused landscape that's shown on the right. That captures the overall defocus as well as the tilt and sometimes curvature of the ice. But when junk is present in the micrograph, sometimes CTF estimation can be substantially ruined by it. And that means you might throw away this micrograph and all the particles that are in it, or the particles in this micrograph may get assigned a CTF that's substantially incorrect, and that makes its way downstream into refinements. This kind of effect is particularly challenging with hexafoil grids, which people are quite excited to use because of their potential for reducing beam-induced motion. Hexafoil grids have much smaller holes and are challenging to process for a few reasons, but CTF estimation can become severely affected by this issue. So a new improvement we've been working on, not yet out in CryoSPARC, is to make use of our junk detector masks in the entire pre-processing pipeline and beyond, to mask out junk so that it's ignored. And this works, but it does take quite a bit of special attention to how to handle sharp edges of masks, masked regions, normalization, etc. So I have some preliminary results to show. So first of all, this is an example from Empire, a hexafoil dataset. When junk is present and not accounted for, CTF estimation is quite poor, and the tone rings are not detected or correctly modeled. If we mask away the junk, we can now recover much better defocused landscape of the good region and tone rings that go to much higher resolution. Across this data set, 2,000 micrographs, without any modifications, just with standard CryoSPARC as it is now, if you were rejecting micrographs that got a worse than 5 angstrom CTF fit, you would throw away about 57% of the entire data set. After junk masking, only 20% is rejected, and many more micrographs are retained because their CTF fits are much better. The effect of junk masking is also measurable in further downstream steps, for example, in the micrograph denoiser. So on the left is a filtered micrograph. The middle is the micrograph denoiser default. And on the right is the micrograph denoiser with junk masking. So the denoiser during training uses the junk masks to only look at parts of the micrograph that are actually good. And that makes a huge difference. This data is Aldelay's data that was shared with us from Dr. Talia Levitz at Harvard. So junk masking makes it possible to process hexafoil datasets much more easily, but also works for other kinds of junk like large ice crystals, ethane contamination, etc., or cases where particles are often close to hole edges. For example, particles that like the thicker ice that's near the edge of a hole. In fact, one of the most important downstream effects that we found so far with junk masking is that we can actually get closer to hole edges and extract particles that would otherwise be thrown away without issue. So these are, again, preliminary results, but on the same Aldelay's data set, we found that processing the particles normally, this is normally with junk masking in CTF estimation and the denoiser, but extracting particles normally, we get a result that's pretty good, decent resolution. Map quality is okay, but a little bit smeared, and the orientation distribution CFAR scores are not great. Instead, if we extract the exact same particle locations, but use the junk masks to fill in particle boxes that would otherwise contain junk with random noise, we can actually align many of the particles much better. We get a slightly improved resolution, but the map quality is substantially improved. CFAR score is substantially improved because of this change. We've also seen some other cases where particles near the edges actually have different orientation distributions than particles in the center of holes. So for larger particles or more challenging data sets, this might be an important effect. Like I mentioned, this junk masking stuff is not out yet, but it is coming soon. So making pre-processing work more robustly is actually part of a larger story that we've been working on for the past few years for enabling practical automation of repeat target single particle cryo-EM processing. In the repeat target scenario, the goal is to solve multiple structures that are related. So for example, this could be the same target with different ligands, different antibodies, maybe in multiple conditions. For example, time-resolved cryo-EM, where you may collect a series of data sets that have the same target, or other more exotic sample preparation techniques, synthetic biology, etc. We've been looking for an approach that can work with challenging targets, so things that are small membrane proteins, things with flexibility, disorder, proteins that are not always happy on the grid, that work on challenging data collections where the data collection itself was imperfect, number of images is huge, signal to noise ratio is low, many of the particles are not intact, contaminants, preferred orientation, etc. Basically, all the things that make single particle challenging from time to time. We've been systematically working towards this goal, looking for an approach that can reach equal or better than manual processing, equal or better map quality than manual processing, which is a tall order, but it's really necessary for an automation approach to really help single particles scale further. So the goal is something that can do this, but is also fully hands-off, meaning that once it's set up for a particular target class, it's fully automated. And an approach that is also simple to generalize to new target classes, meaning that there shouldn't be a lot of custom, finely tuned parameters that need to be figured out again and again every time we're looking at a different type of target. So we've been working on this problem for a while. We shared some initial results back in 2024. Since then, we've been developing and releasing some new methods and tools in every CryoSPARC version along the way. A few months ago, we posted a preprint where we demonstrated and gave a lot of details about a general automation approach that we've developed that can solve, for example, a wide array of GPCR structures equal or better than manual processing. I'm going to talk through some of the details of those results. And I'm also very excited to share some newer results on a different target class. And I think some of the takeaways from this work are, of course, automation, but also some of the strategies that we found to be robust enough that even for manual processing may be a good idea for people who are thinking about modifying their own workflows. So GPCRs are a key drug target, and Cryo-EM has been a huge enabler of GPCR structures to date. But processing GPCRs is notoriously difficult. We gathered 21 GPCR datasets to construct a challenging test case for automation. These are all different species, data from different microscopes, different labs, different sample preparation grid types, cameras, etc. Nearly all of these datasets have some kind of ligand bound. We divided the datasets into 4 target classes based on whether or not they had a Fab and whether they were the active or inactive states of the GPCR. So there are 3 active state target classes that have a Fab in a different location each and 1 inactive state. Our goal was to find an automation strategy that we could set up once for each target class and would allow fully automated processing of all the other datasets in each class. We went through a lot of concepts, iteration, experiments. The approach we finally developed makes use of limited prior structural knowledge about the target class, and that is in the form of a low-resolution 15 angstrom reference map representing our target class and some approximate particle diameters. The reference map does not need to be very precise, as our results show, and it can come from a previous Cryo-EM data collection or processing of a single exemplar, for example, from the target class, perhaps AlphaFold or other sources. So what we did is we processed 1 data set from each GPCR class and used a 15 angstrom version of the map we got from that to automate the rest of the processing. We generally followed best practices that have been amassed over the years from manual processing, but we did find a few key areas that needed special attention to really work in an automated context so that we could actually get high quality results, but remain robust to variable quality data, rare low contrast views, and be able to curate high quality particles from very large sets where many of the particles were broken or denatured. So the final strategy that we came up with, I'll go into some details, works across all of our GPCR datasets, including the active and inactive states. And the inactive state is even smaller than the active state. Some of the key points are that the denoiser and junk detection that I mentioned are quite important, particularly for simplifying pre-processing and curation, but also making particle picking far more robust. Particle curation, we found, is far better to be done entirely in 3D, so no 2D classification for curation. which is actually crucial to curating without losing rare views. And we also automatically perform multiple refinements and then select the best one to optimize for microscope parameters, CTF refinement parameters, et cetera, that are necessary to achieve the highest resolutions. So in terms of pre-processing, motion correction, CTF estimation, of course, are well established and automatic at this point. Using the denoiser and junk detector, like I mentioned earlier, is quite important because denoising makes the signal from particles stand out strongly and makes particle picking far more robust, so that we can automatically filter particle picks without any user intervention based on just the picking scores through a clustering technique, and end up with very good initial picks that we can also use the junk detector to discard problematic micrographs and picks that are too close to junk. In the automated workflow we developed initially, we started with blob-based particle picking on denoised micrographs, and then improved that using 2D classification and automatic selection of 2D classes to produce templates that were then used for template picking. This worked quite well, but in our more recent improvements, which I'll talk about in a bit, we found that actually picking directly using 2D templates generated from the low resolution 3D reference map actually works just as well and is far simpler. And especially on denoised micrographs. So that avoids the need for 2D classification now entirely. Curation is actually the hardest part of processing data automatically because particle picking can yield millions of particles from micrographs. Typically, most or even sometimes 90% of them are not good particles, and including them in downstream steps ruins map quality. They might be broken, denatured, misaligned, thick ice, contaminants, etc. So particle curation, we start with a technique that's known in the community as decoy classification, which many people use already for manual processing. The idea there is to use our coarse 15 angstrom reference map as one input, along with several junk density maps that are either input or automatically generated into a CryoSPARC heterogeneous refinement. That heterogeneous refinement takes all of the particles, regardless of their quality, and simultaneously aligns, refines, and sorts them between these classes. So the particles that end up in the intact class, and we already know which one that is because we use the reference, we can consider those to be good particles, and the rest we can discard as junk particles. Decoy classification is highly robust to junk particles, even when there's only 10% or less good particles in the input set. But alone, we found that it can't actually curate well enough to get great quality refinements if you only use this step. That's where the next phase of curation comes in, rounds of multi-class ab initio reconstruction, followed by rounds of heterogeneous refinement. The key here is that in multi-class ab initio reconstruction, that's on the top right, we are now generating density maps directly from the data with no input references at all. And so any intact structures that come out are of the particular target that's in the actual data set, and the junk and other classes are also data set specific. So that followed by heterogeneous refinement, where now in heterogeneous refinement, at least one or more of the density maps that come out are intact versions of our particle. Selecting just those particles and those classes yields far better curation. We can select the classes automatically, either using the 3D reference map or just based on simple statistics like resolution and orientation distributions. So the particles that come out of this curation can go into high resolution refinement. For this, we run multiple non-uniform refinements in parallel, each one optimizing a different set of CTF parameters so that we can automatically determine the best selection. CTF refinements, these refinements, the non-uniform refinements, produce high quality density maps of the target molecule, but GPCRs and many other targets are sometimes dynamic, and GPCRs are notoriously slow, especially in the receptor domain. So to improve map quality further and do that automatically, we also perform a local refinement by aligning our particles and reference and map up to this point to the 3D reference that we have and using a mask that's also aligned to that reference. With that local refinement, that produces our end-to-end automated best maps that we can get. So we can take a look at how this worked across 21 GPCR test datasets. These are just a few examples. First of all, the automation approach is successful in completely hands-off processing on all the datasets. In nearly all cases, the automated results meet or exceed deposited resolution, but more importantly, the map quality is similar or improved in many cases, especially for the ligand. To summarize those results, this chart here shows on the left side resolution, on the right side CFAR, which is a measure of map anisotropy. The empty circles are manual process deposited results. The filled dots are our automated results. So in 17 out of 21 cases, our results meet or exceed the resolution compared to manual processing. In the other 4 cases, the results are within 0.2 angstroms. Sometimes the resolution improvements from automation are as great as 0.7 angstroms, but of course that depends on how the manual processing was done as well. Importantly as well, automated processing improved the CFAR score in 12 of the 15 cases where the depositors had deposited half maps. So this goes to show that our automated particle curation did a good job of retaining rare and low contrast particle views, and that's what improves the orientation distribution. In the end, what's most important, of course, is the protein and ligand map quality. So I'll show just a few examples where we saw substantial improvements. This first case, you can see the receptor domain map quality is much improved, and we're able to model more of the protein and side chains. The left is the published manual processing, the right fully automated processing. Second case is an example where manual processing yielded a map where the ligand could not be seen, whereas automated processing revealed it. That's the green atoms in the middle. And the third example is a case where the nominal resolution of the automated processing matches the manual processing, but the map quality is sufficiently improved to model more of the ligand. Okay, so these are great results, but we've only shown results on one target class, GPCRs. Within that target class, we had 4 subclasses. What we wanted to do next was see what happens when we apply the same approach to a different target class. So I'll talk about some more recent results applying this approach to the cyclin-activated kinase, which is a promising target for cancer therapeutics. There are 9 data sets that were collected in a very impressive work that was published in 2024. Fortunately, the authors deposited these to Empire. There's an APO data set, 8 data sets with different ligands bound to the same binding site of the CAC protein. The data was originally processed by the authors in a combination of CryoSPARC and RelyOn, and they produced maps with very high nominal resolutions, around 2 angstroms in some cases. However, the data sets are quite challenging, primarily because of strong preferred orientation. And because of that, the maps are highly anisotropic and map quality is degraded in some cases. So this is an example of a deposited map. You can see that there is strong streaking, and in many areas, the density is broken despite high resolution information being present in some directions. So we started with the same automation strategy that was working well for GPCRs, and the essential steps have remained exactly the same, but we've just further simplified it and made it more robust. So the workflow still requires only limited prior knowledge about our target class. In this case, we used a density map from processing the apostate CAC dataset. So we just processed that one dataset and used its final density map. We simplified particle picking by generating coarse, low-resolution 2D templates from the 3D reference map that we had and picking on denoised micrographs, which means we no longer need to do any 2D classification or any selection of 2D classes. All particle processing is done entirely in 3D. And within the workflow, alongside the curation steps that I mentioned, we also added steps to rebalance particle orientations and filter particles by the per particle scale metric, which is quite useful. These 2 changes made the workflow more robust to preferred orientation issues generally. Aside from the input reference map, we didn't actually need to change any other parameters of the jobs between this workflow and the one that worked on GPCRs. In fact, we even used the same exact junk volumes for decoy classification. When we look at the results, we can see that on this very different target class, automation worked actually quite well. And in fact, we obtained final maps that appear better in quality than the published maps in many of the cases. And all processing of the ligand-bound datasets, again, completely hands-off. Here's an example of one of the ligand-bound datasets. On the left, the deposited map. On the right, the fully automated processing. By eye, you can see that the automated map no longer has those anisotropy artifacts. The density connectivity is improved. Sidechain ligand density are clearly resolved. So this was looking great when we saw it, but to confirm the quality of the automated maps, we wanted to measure a series of map quality metrics. First of all, the same 2 we had before. On the left, resolution. On the right, CFAR. Our automated results are all better than 2.2 angstroms. Nearly all are within 0.1 angstrom of the deposited map resolutions. But more interestingly, on the right are CFAR scores. The deposited maps all score below 0.2 and many of them near 0, meaning that there's severe orientation bias and the map can't be trusted in certain viewing directions. The automated maps all score 0.4 or better, and most of them are near 0.5, meaning that the maps are far more isotropic and interpretable, And while there's still an orientation preference in the data, it's no longer adversely affecting the map quality. Once we got these automated maps, we wanted to, aside from these metrics, we wanted to absolutely confirm that the map quality was really better from a model building perspective. So for every data set, we rebuilt an atomic model, redocked the ligand, remodeled the waters. And what we found is that on a variety of scores that we could test, the automated maps were better than the deposited manually processed maps. Again, here, empty circles are the deposited maps with the deposited model. Filled circles are the automated map and our rebuilt model. So on map to model cross correlation, EM Ringer score, mole probability score, clash score, and the number of waters that could be modeled, the automated maps were substantially better. This video just shows the ligand binding region, the pocket, across the automated density maps. So we were really excited to get these results, able to process all the data sets here fully automatically, each one taking on average only 19 hours to process on a single 8 GPU server. These are very recent results, and the details of all of this and the CAC automation workflow, et cetera, will be out soon. In general, we're very excited about the prospect of repeat target automation because of the new kinds of biological questions that it may help to make easier. Of course, drug discovery, but also things like time-resolved cryo-EM, synthetic biology, and other applications that we haven't seen yet. So if you have any repeat target data sets that you might be able to deposit or share, please do reach out. They will make all of this story really exciting. Another quick side note, we recently released CryoSPARC v5 in a public beta a couple months ago. It has a huge number of new useful features, so please do take a look, upgrade when you can. It includes a major overhaul of 3D masking and refinements, new use of per particle scale metrics, improvements in CryoSPARC Live, a new dashboard and comparison view, improvements in 2D classification, 3D classification. The UI improvements are particularly helpful. They allow you to quickly compare multiple jobs, scrub through different iterations of different plots, compare plots between jobs, and so on. One final shout out, high resolution ab initio reconstruction. I think we're going to hear about this a lot more in Ollie Clark's talk later today. I just wanted to shout it out because it's a really exciting story. Ab initio reconstruction that we developed originally back in 2017 in CryoSPARC was designed really for solving density maps from scratch just to medium resolution, and all of its parameters were tuned for that purpose. So we were really excited and surprised to see the amazing results that Ollie Clark and his group have had running high resolution ab initio by changing some of the key parameters, letting it run for many more iterations. And I'm sure we're going to see many more of these results later today. But it's really exciting because it massively improves map quality for small particles, maybe pushing the limits of what we can do with single particle cryo-EM. In version 5, we actually added a new job type called ab initio refinement. This is a single class ab initio job that maintains a gold standard split between two half maps. And so it can be used for downstream validation correctly. So definitely check that out if you're interested. And finally, just a shout out to our guide and discussion forum, which are great resources for anyone trying to learn single particle processing, learn how to use CryoSPARC, or connect with the community, ask questions. Our team also monitors the forum very regularly, so it's a great place for community engagement. Thank you very much. Any questions?